jump over to here. We should be good to go. Okay, so the story with reproductive system, interestingly enough, is that this system, of all the systems of the body, is the one that is least concerned with homeostasis, right? Almost every other system of the body that we've been learning through 202 and 201 is all about maintaining this internal environment, maintaining this balance of some aspect of the body, whether it's oxygen concentration or glucose levels or fluid levels or whatever. It's all about maintaining the body itself, whereas this system is more about creating another body. Right? So there are some effects when we talk about the hormones, when we talk about the male and the female sex hormones, there are some metabolic effects of those, etc. But for the most part, this system is geared towards making a new individual. So it doesn't necessarily contribute that much to the body that it's in. Interesting. Okay. So when we are looking at the system, why is that pulled out? Let's put it back in. Between the two aspects of it, the two types of reproductive system, that's something that we have to deal with this time, right? All the other systems, we just go, oh, urinary system, this is kind of how it works. Respiratory system, everybody's got two of these lungs, etc. In this system, right, it's like two different systems in one. So it does take a bit more time, but we have a bit of time to be going through it. Overall, though, between the male and the female reproductive system, there's still some overall general functions for it. So, for instance, we're talking about making sex hormones, like we talked about before, and making gametes, making the sex cells that can make a new individual, and then kind of protecting those sex cells, supporting them, transporting them as well, to be able to potentially get them to join at some time to make a new individual. Okay? So we're definitely going to see, like we have before, like when we were doing digestive system, etc., we're going to see a pathway that these different gametes are going to follow. We're going to see different organs that will contribute to the main street of that pathway that they pass through, but also a number of organs that maybe produce secretions or have some sort of supportive function as well. Now, when we look at this system, the primary sex organs, which we're going to start with, are potentially not necessarily what you would think. So the primary sex organs are going to be the gonads. So the gonads are going to be the testes and the ovaries, right? So the primary sex organs are those that actually make the gametes. Because all of the rest of the glands and accessory organs and things like that certainly are important, but if you don't have the gonads making the gametes, then all of those other organs are not going to be able to make a new individual, right? So the primary sex organs are going to be the gonads. So they produce the gametes as well as the sex hormones that we're going to be talking about. Everything else counts as an accessory reproductive organ. So ducts, glands, external genitalia, etc. And as we go through, we're going to start with the male reproductive system, then we'll switch over to the female reproductive system for a couple of reasons. Number one, to a certain extent, the female reproductive system is a little bit more complex, because we've got those two cycles that we were hinting at before. We have the ovarian cycle, and then we have the uterine cycle, which have to be coordinated with one another under the influence of hormones. And so we have to understand how that works. But then also, it makes sense to do the female reproductive system second, because then we will graduate right into development after that. So it'll be a kind of a smooth transition that way. Okay. So like we said, the sex hormones are going to be an aspect about this as well. For the female reproductive system, helping to coordinate the menstrual cycle and the potential reproductive cycle on a monthly basis. But then it also contributes to sexual behavior and sex drives, as well as some growth and development and metabolism for other areas of the body. So they have a number of functions, but primarily, interestingly enough, these sex hormones work within the reproductive system to coordinate the reproductive system's activities overall. Okay, so male reproductive system. We have the male gonads, our primary sex organs, that are going to produce sperm, and they're going to lie within a sac that's called the scrotum. And so in this way, of course, we're going to highlight differences and similarities between the male and the female. Interestingly, right, the gonads, as you've already seen, because we've done anatomy in the lab, the gonads for the female are in the body cavity, right? The ovaries are within the pelvic cavity, but the gonads for the male are outside of the body cavity. And we'll talk about the reason for that. So they're located in the scrotum, and they are going to create the sperm, and then they're going to be delivered to a number of different sections of tube along the way that are each going to kind of contribute. So we're going to have what's called the epididymis. So if you remember again from lab, that's kind of a comma-shaped organ that sits on top of the gonad, on top of the testis. We have the ductus deferens, which is also called the vas deferens, which is going to transport from the epididymis into the body cavity around the sides of the bladder, bring it together in an area that we call the ejaculatory duct, where one of our first accessory glands are going to put in their secretion. And then we're going to jump into the urethra. And as we were thinking about it in terms of the urinary system, we know that the urethra in the male actually has 3 sections. So the first section is going to the prostate gland, which is just inferior to the bladder. And then we have a very small section that is going to be called the membranous urethra that passes through the body cavity wall. And then we're going to have the penile or spongy urethra after that. So that's going to be, in essence, kind of part of our tracing, where we're going to see again kind of the possibility of questions that ask you to trace a gamete from the point that they're produced until they're released from the body. So we're seeing a number of the structures that we would need for that tracing, but we'll go over that again later as well. So again, like the digestive system, we've got a main street where things are going to be passing through, but then we're going to have these accessory organs dumping in along the way. And again, think back to when you were doing the anatomy in the models and things like that in lab. Seminal vesicles, so these are one of the first glands that dump in. There's 2 of them on either side of the bladder that dump in their secretions. The prostate gland, as the name indicates, is also a gland that the urethra passes through. And then we have the vulvourethral glands, which are one of the last glands along that pathway. So again, in essence, they'll all be contributing secretions that will then kind of mix with the sperm that are being delivered from the epididymis directly, and they'll support them in some way, help in transport, help to protect them, help to give them nutrients, things like that. So we'll talk about how each one helps along the way. So this mid-sagittal view should be pretty familiar in terms of what we've seen in the lab as well. So we're starting down here. Here's our primary sex organs. There's our testis sitting within the scrotum. Here's our epididymis sitting on top right there. So the epididymis, as we'll talk about coming soon, is going to be storing and allowing the sperm to mature and kind of activate. They become more active when they're transferred from the gonads into the epididymis. So the epididymis is where they're going to be kind of activated. They're going to be concentrated there. And then, with emission and ejaculation, they'll move from the epididymis, and here's our tube. So this is our vas deferens, or our ductus deferens, and this is going around the lateral sides of the bladder, like we saw. And then this area along here gets a little bit thicker. This is called the ampulla of that ductus deferens, and we're passing by one of our first accessory glands. That's the seminiferous tubule that we have right here. And then as it comes into the prostate gland, there's a separate little channel here from the side. That's the ejaculatory duct. So there's 2 ejaculatory ducts where these 2 vas deferens or 2 ductus deferens are transporting sperm from each side. And then those dump into the singular prostatic urethra that we see right there. So the prostatic urethra is just that first little section that goes from the bladder inferiorly. So in the male reproductive system, we see that the urethra is going to be dual function. It's going to be transporting urine from the bladder, but it's also going to be transporting semen, which includes the secretions from these glands, plus the sperm inside. So there's our prostatic urethra. This little section right here, once we leave the urethra, or once we leave the prostate, and we're transitioning through the body wall, that is the membranous urethra. That one's a little bit tough to see on some of the models, like the purple black, but you can see it on the torso model. So just that little section as it passes through the body wall is the membranous urethra, and then the rest of this is the spongy or penile urethra. And we almost missed our last gland right here, there are paired bulbourethral glands that dump in here right at the beginning of the spongy urethra. Now when we look at it, the spongy or penile urethra, the penile urethra, that makes sense, right, because it's going through the penis. But at the same time, in terms of the spongy urethra, the reason that it has that name is that there are actually 3 channels of erectile tissue that go through the penis. And there's one that encircles the urethra. So in essence, like there's kind of a dividing line along here. This is called the corpus spongiosum. This is the spongy erectile tissue here that surrounds the urethra. So all of that is called the corpus spongiosum, the spongy body, if you will. And then on the dorsal surface, there's an additional 2 channels of erectile tissue up here, which are called the corpora cavernosa. So there's 2 of them. A single one's called the corpus cavernosum. So the reason that that urethra is called the spongy urethra is because it passes through the corpus spongiosum. So you can use either term for it. It would be the penile urethra or the spongy urethra, that last one. Now the spongy urethra empties out, right? So it transports our urine as well as our sperm. And when we look at that corpus spongiosum, you can see how it expands distally. So it expands to make the head of the penis, which may or may not be covered with a prepuce, with the foreskin like that. And then we have an external orifice that leads to the outside. So again, a series of tubes, or in essence, one tube, one main street, that has these accessory organs along the way that we'll talk about why we have these different ones. Why do we have seminiferous tubules, and a prostate gland, and bulbourethral glands? How do they contribute each in terms of the semen? Okay, so we said we start out this talking about the gonads, talking about the testes. Now we'll get into a little bit more detail about that coming up, because obviously we're going to have to spend some time there talking about how those sperm cells are created within those seminiferous tubules, those tubules that we saw in the microscope. If you remember, we looked at those previously. But in essence, the gonads are going to be sitting within the scrotum, like we said, outside of the body cavity. And the main reason for that is that the process of creating these gametes, so generally that's called gametogenesis overall, right? In essence, kind of sperm being born, or gametes being born, it's going to be gametogenesis, or you can call it spermatogenesis if you want to say more specifically for the male. That process and the enzymes that are used actually has to occur at a temperature that is lower than body temperature. So in order to regulate that, the gonads are located outside of the body cavity. Core body temperature is about 100 degrees Celsius. And in fact, spermatogenesis needs to occur at a temperature about 3 degrees Celsius cooler than that. And so having it outside then, and because these sperm cells are created almost continuously from the teenage years, from sexual maturity until basically the end of life, they're going to be made all the time, and that process has to be at a lower temperature, and so that's going to be very important in terms of keeping those gonads outside of the body cavity. Now, when we look at it, in fact, there are muscles associated with the scrotum as well. So we can see, here's the gonad right here. Here's the testis. There's the epididymis sitting on top. And you can see not only do we have the ductus deferens heading up, but there's a series of blood vessels as well as nerves and lymphatics that actually go down into the scrotum as well. So those are all wrapped together in what's called the spermatic cord, as you can see on this side and the left side. So those are all bundled together in the spermatic cord. So it's not just that ductus deferens, but a number of vessels, etc., that are going down there. But in addition to that, we have muscle that help to regulate temperature as well. So we have what's called the cremaster muscle, and then there's also, where's our other one? Dartos muscle that we see right here. So there's 2 sets of muscles that also contribute to regulating the temperature of the internal environment, at least in terms of the gonads themselves. So we said it needs to be 3 degrees cooler, but in fact, that temperature is going to vary based on how warm the body is or how cool the body is, or the external environment. So that intra within scrotal temperature is kept constant by those two sets of muscles. So the dartos muscle, which is more superficial, can wrinkle the scrotal skin, which makes it kind of tighter to the body, closer to the body. And then the cremaster muscles, which are more what you see here, kind of wrapping the spermatic cord as well as wrapping the testis itself, are bands of muscle that actually elevate the testes. So elevate the testes when it's cold, right? So when it's cold, then it can be too cold. It can be beyond that 97 degrees. And so we need to bring them up closer to the body to stay warm. But then when it gets warm, basically those muscles can change so that we can descend the testes and get them away from the heated up body to make sure that they stay cool enough as well. So there's kind of this changing dynamic in terms of these muscles to regulate the external environment of the gonads. Okay.
Good question.
Yeah?
So with the cremaster, the skeletal muscle, is there voluntary control of that?
Yeah, that's a good question. So it's not really voluntary control, right? So the guy's not going to be like, let's bring him up. But, you know, if we think about like the diaphragm as well, a diaphragm technically is also skeletal muscle, but it's being controlled on a subconscious level. So the same thing in terms of like the cremaster muscle. So it counts as skeletal. It's not smooth, it's not cardiac, but it's definitely controlled at a subconscious level. Yeah. And in fact, it can be influenced by the sympathetic nervous system and things like that as well, right? So if there's an emergency, then things can tighten up. You don't want things swinging around and, you know, endangering your ability to make new individuals, right? It's pretty important. Yeah. Okay. So when we're looking at the testes themselves, they are encased within like a peritoneum as well as a fibrous capsule. So to a certain extent, it's kind of like when we were looking at the heart, right? There's a membrane that surrounds it as well as a connective tissue bag. When we look inside of the testis, we find those seminiferous tubules. And those tubules are coiled up into small lobes, which we call lobules. So there's 250 to 300 different lobules, different little regions inside of the testis, each one containing 1 to 4 seminiferous tubules. So when we saw them in the microscope, there are all these cross sections, right? These donuts, these cross sections of tubes that we were seeing. And so there are lots of those divided into these areas that we call lobules. And we'll see that more coming up as well in terms of diagrams. Okay, so we saw those seminiferous tubules, of which there are lots of them. So just like we saw with the small intestine, lots of coiling, lots of going back and forth gives us a lot of surface area because we know within the walls of those seminiferous tubules is where we are making the sperm. So the seminiferous tubules are producing the sperm, and we'll talk about the different stages a little bit more in detail. And all of those tubules then, in a lobule, then come together in kind of a drainage point that's called the tubulus rectus. So rectus means straight. So these straight tubules are going to be taking it to what's called the reedy testes. So let me show you, we're seeing a lot of terms here. I think it's better to have a diagram coming up like this. So here's the testis that we see down here. This is a lobule like this. So we've got like 250 to 300 of those lobules, and then within each one, 1 to 4 seminiferous tubules. And you can see how all of this is going to drain into this straight area along here. And then that goes into this collection area here, which is going to be the reedy testes. So we're gathering them to a certain extent, kind of like we saw in the kidney. All of these different nephrons throughout the kidney that come together to the collecting tubules. Collecting tubules bring down together to the renal papilla. Those are collected to the minor calyx, to the major calyx, etc. So we got to bring all of those tubules together. So that happens here, the reedy testes. And then the reedy testes is going to send it to our epididymis. So up here, all of this is epididymis. And so you can see that there's a number of coiled tubes of the epididymis as well. And we saw that in the microscope. Now the epididymis, the volume of the epididymis is much smaller than that of the testis. So as we were saying, not only do we do activation of the sperm, allow them to become more mobile in these kind of last maturation stages, but we do a concentration of it as well. All the production of sperm that are occurring in these different lobules kind of have to be washed out. You saw we have more mature stages that are in towards the lumen, and then we saw those tails extending in towards the lumen. There's some fluid that basically washes them out so that more can be produced and released. So it gets washed out, and then it gets sent up to the epididymis. And the epididymis, some of that fluid then gets reabsorbed so we can condense it and pack it into the epididymis for storage that we see there. And as we mentioned, during emission and ejaculation, that's where it's going to be coming from. It's going to be coming from the epididymis. So in a way, it's a little bit like when we talked in the digestive system, how the liver makes the bile, but the gallbladder stores the bile. So in this case, right, the testes and the seminiferous tubule make the sperm, but they're actually stored in the epididymis. So during ejaculation, they're coming from the epididymis as opposed to directly from the testes themselves. Okay. So on the opposite side of this whole pathway, we have the penis as well. Now we're going to be talking about the glands and things like that as we go through. But a lot of times when we get into the system and we talk about the primary sex organs, right? People think, oh, I get it. Primary sex organs are going to be the penis and the vagina. But as we said, it's going to be the gonads instead. So the main function for the penis is to, as we saw, the urethra going through it is going to be allowing urine to go through it, but also to be transporting sperm and depositing them within the female reproductive tract. And so the critical aspect about that is to be able to deposit them into the vagina where they can then migrate up to the cervix. We know the cervix is the opening for the uterus down to the vagina. Migrate up to the cervix, enter the cervix, then migrate up that uterus and start heading towards those uterine tubes. So that's an interesting thing that we'll talk about as we flip and we get to the female reproductive system. All those sperm are being deposited in one area, and they can only get so far, right? They're deposited into the vagina. They have to propel themselves the rest of the journey. And so they have to go into the cervix, up into the different areas of the uterus, like we said, right? There's the isthmus, the skinny part, then there's the body, then there's the fundus. They're going up all the way through that. And then they have this big wide junction where they have to figure out which way they need to go. Because on a monthly basis, it's only one of those ovaries that actually releases an egg. So if they start heading off towards this way, they just did their whole life's journey, and there's nothing at the end of that pathway if they make a wrong turn, right? So it's kind of interesting to think about. And that actually plays into the fact, like we said, that the fact in the male, it's like a numbers game, right? Millions and millions and millions of sperm that are released in an ejaculation, because not all of them are going to go the right way. Not all of them are going to be fully functional, like swimming right, etc. So it's just a numbers game that they need that many so that at least some end up in an area where that egg might be released. Because that's the goal of this system, right? It's not something that we, right, as conscious beings, we're not like, yep, we're going to do this every month. We are definitely not going to do this every month, right? Even if it's achieved, then you have 9 months, there's a few months afterwards at least, before things get up and running again in terms of the ovarian cycles and things like that. But each and every month, that's what the female's body's preparing for. It's like, this might be the month where we get pregnant. So it has to prepare for that each and every time. And the male gonads have to make sure that there are viable sperm that are ready to go all the time, just in case. Yeah?
How long do they last? Like, how long do they live before they just start dying off? 3 to 5 days. Oh, whoops.
What's that?
Never mind, Michelle knows everything.
What did she say?
I said 3 to 5 days.
Yeah.
I was.
So it's on the order of days, to be sure, like outside of the body. So they're maintained internally, like we said, transferred to the epididymis and stuff like that. But then once they're transported out of the body, they are being stored inside the body. We're packaging things together with them from those accessory glands that are going to help them and protect them, etc. But realize that when they exit the body, now this is a hostile environment to a certain extent, right? This is not supportive anymore in terms of temperature, in terms of chemicals and things like that. And even the internal environment of the female is relatively hostile as well, right? Just because there's an immune system there that is looking to attack anything that is foreign. So there's a number of these sperm cells that actually get taken out by the different protective measures of the female reproductive system. So again, that's another reason why there's a numbers game, is that if we're even going to get one successful one to get up there to fertilize that egg, there are so many that are going to not make it along the way that we have to have that many to just increase the chances of that possibility. Okay, again, the main idea with the penis then is to deliver it, to be able to deliver it into the vagina itself. So we've got that erectile tissue, as we mentioned, the spongy network of connective tissue, as well as smooth muscle that has lots of different spaces in it that can accommodate blood. So we'll talk about that in terms of how that happens in terms of the erection and again, the deposition of the sperm into the female reproductive tract coming up. Now also, in terms of tubes and things like that, we have the urethra, but if we're working our way back, we also have, as we mentioned, the ductus or vas deferens and the ejaculatory duct. So this is where it's coming from the two sides, because we have two testes, two sides that are bringing these secretions as well as the sperm together. So the vas deferens and the ejaculatory duct are going to be running, again, from the epididymis. Vas deferens is going to be running into the body cavity. There's a small area where it gets a little bit larger called the ampulla, and that's where it kind of comes right near to the first of our three different accessory glands called the seminal vesicle. So as those two merge, that gives us our ejaculatory duct. That is going to continue to be able to transfer the sperm around this pathway that they're having to go, around the sides of the bladder, going through this pathway. So the vas deferens is basically going from the epididymis into the ejaculatory duct. And this is the place where, if we're talking about a vasectomy, where we're trying to prevent those sperm from getting where, evolutionarily, where they're supposed to go, this is the way that they cut off the path. So they'll go in to that spermatic cord, and they will ligate, they'll cut, and then tie off the vas deferens. So in that way, there's no way sperm and the sex hormones can still be produced by the gonads. They can still be transferred to the epididymis, but there's no place for them to go. So they can be produced, they'll be produced at a much lower level, and in essence, with no place to go, they basically get reabsorbed. So it's kind of a recycling type of process to them, that in this way they have no place to go for as long as that vasectomy is still active. It is possible to reverse that as well, and then kind of, if you will, free them up. But during that time, they're just going to be reabsorbed instead. So, sections of tubes, as we mentioned, we have our vas deferens, we have our ejaculatory ducts. Here's our urethra that's going through a section of it, going through the main section of it, going through the penis. So we have the first section going through the prostate, the second and smallest section going through the body wall, going through what's called the urogenital diaphragm, and then the longest section, the spongy or penile urethra, going through that corpus spongiosum. So that urethra, like we said, is going to be dual function in the male. In the female, reproductive and urinary pathways are totally separate, but in the male, they're shared. Now, of course, happening at different times, and in fact, there are bands of smooth muscle that can regulate that to make sure that they don't go at the same time. So, in fact, when the male reproductive system and the male emissions and kind of transporting sperm is eminent, there are bands of smooth muscle that will keep any urine that's in the bladder from going through the prostate, from moving on down. So there's a band of smooth muscle right there that helps to regulate it. Just like we saw with the digestive system, to make sure things will stay at a certain section when we need them to, or allow them to pass through for a certain period of time. Okay, so the tubes are just about getting it from one place to another. Vas deferens from the epididymis into the ejaculatory duct picking up the secretions from the seminal vesicles, dumping them into the prostatic urethra, and then the prostatic to the membranous to the spongy urethra and out. So what about these accessory glands? How are they contributing? Why do we have so many of them? So the seminal vesicles, of which there are two, that's the plural aspect of it, are on the posterior body wall, posterior wall of the bladder itself, and they secrete about 60% of the volume of the semen. So semen itself is going to be the sperm cells plus the secretions. Some of the secretions for washing the sperm through the vas deferens, but also additional secretions that support the sperm in some way. So in this case, what we have is a number of things that will support, like we said, support and or protect the sperm cells. So you'll notice, what's that? Fructose. That's sugar, right? So these cells are particularly active. We've seen, when we looked at the seminiferous tubules, how their morphology actually changes, right? They start out as a generic-looking cell with a big centrally located nucleus and lots of cytoplasm, etc. But as they develop and as they get closer into the lumen in that seminiferous tubule, they become very, very streamlined. So they're losing a lot of the cellular machinery to support themselves, and it's more about just being able to propel themselves to the potential site of fertilization. And so with that, we have mitochondria that are going to be generating, in that mid piece, if you remember, that are going to be generating the energy to rotate that flagellum so that they can propel themselves. But they don't have a lot of cellular machinery to be able to, again, generate a lot of energy, etc. And so actually bathing them in sugar makes sure that they have some sort of energetic resource available to them right there. So fructose, see ascorbic acid in there. Interestingly, there are coagulating enzymes in there. So the deal, right, is that this whole pathway is about transporting the sperm cells through these different tubules, getting them into the urethra so that potentially they can be deposited into the vagina, right? But when they're in the vagina, the whole idea, again, evolutionarily of this system, is to get them to join the egg. So we have fluid in there, which makes, the sperm able to move, but it's also possible that this semen could actually just move out of the vagina, and now that's working against what the sperm are trying to do, right? The sperm are trying to go up, and this secretion is coming down. So that's not going to be very adaptive in terms of successfully making a new individual. So this enzyme from the seminal vesicles actually temporarily coagulates the semen. So it makes it more viscous so that it can stay within the vaginal cavity. So it can adhere to the wall for a period of time, giving them more of a chance of staying there on their pathway up to meet a released egg that's coming from the ovary. All right, so we have a coagulating enzyme in there as well called vesiculase, and then there are prostaglandins. We know that prostaglandins are chemical messengers. These prostaglandins, when put into the female reproductive tract, put into the vagina, can actually stimulate smooth muscle contractions within the wall of the vagina as well as we get up towards the uterus. That can, again, support, these contractions can work to move the sperm, again, closer up to the cervix and up along this pathway where they are destined to go. So a number of things that we see coming in terms of the seminal vesicles that are, again, contributing and supporting and potentially protecting those sperm cells. So from there, once we pass the seminal vesicles, as we mentioned, we go into the ejaculatory duct, and then we're going to dump into the prostatic urethra. So we started out, we've got most of the semen in terms of the fluid coming from the seminal vesicles. The other 35%-ish comes from the prostate gland. So the prostate gland, there's only one. There's two seminal vesicles, two vulvourethral glands. There's only one prostate gland. As we saw, that is inferior to the bladder because the urethra passes right through the center of it. In this case, the prostate gland puts out an additional fluid. It kind of comes from all sides and it drops into the prostatic urethra. It has citrate. It has additional enzymes to it as well. Now, it puts out a particular antigen, the prostate-specific antigen. Interestingly enough, the PSA is something that they measure in men to look at the size or assess the size of the prostate. Because the prostate, through time, interesting, it seems like it would be the biggest in, who knows, like your 20s and 30s and stuff like that, where definitely this reproductive system is getting cranked up and being very active potentially, etc. But as you get into 40s and 50s and things like that, the prostate itself gets much larger. So it's called benign prostatic hyperplasia. So it gets a bit bigger, and that can restrict the flow of urine through that prostatic urethra. So as guys get much older, middle age and then much older, it's actually harder for them to go pee because that tube is getting restricted. So it gets harder for them to fully empty the bladder, etc. But we also know that the prostate is one of the areas that can become cancerous, right? You've heard of prostate cancer. And so what they want to monitor is, we know that the prostate itself is going to get bigger as you get older, but a normal amount of growth, so seeing that PSA increase at kind of a general rate through time is okay, but if they see a big spike in that, then that may indicate prostatic cancer. And so that's the most common way that they monitor the prostate now in terms of its growth. The other way that was more common for a long time was called a digital exam. And you're like, oh, digital, like a computer? No, digital exam, right? Because if you look at that mid-sagittal cut, the prostate gland is right next to the rectum. And so in order to be able to assess the size as well as, right, when we have cancerous tissue, it gets bigger, but it also, we see growth in weird ways, right? So it's not quite smooth. You have maybe bumps to it, like tumors and things like that can grow. And so they're trying to feel for if there are those types of bumps or growths on the surface of it. But this way is a little bit more comfortable and is a little bit more effective. So they monitor PSA levels throughout doing blood work, monitor that throughout the later stages, if you will, or the mid to later stages of the male reproductive lifetime. Okay, but one of the other interesting things that I want to mention about the prostatic secretions is that the enzymes that we have in there actually work together with those from the seminiferous tubules. I'm sorry, so the seminal vesicle. So the seminal vesicle, we said, has coagulation enzymes that works to temporarily make the semen more viscous, so that when it's deposited within the vagina, it tends to stay. But think of this, right? So, okay, so we made it more viscous, so the sperm are now staying in the vagina. So evolutionarily, that's a good thing. But what if they're stuck in there, right? So if it gets really viscous, how are they going to get out of there in order to get to the cervix, in order to get to the uterus and continue on their pathway? So for a period of time, that enzyme from the seminal vesicles is active, but then there is a liquefying enzyme that comes from the prostate gland that's kind of a delayed type of activation that then re-liquefies the semen so that the sperm are actually freed so that then they can go on their way. So it's a bit like when we were talking about the clotting cascade, the blood clotting. There's that really quick cascade that got us to activating fibrinogen to fibrin so that we could form a clot. And if you remember, the way that we got rid of clots is there was plasminogen, a different enzyme that took a much slower amount of time to become activated, so that after the fibrinogen has formed that clot and the blood vessel is healed, by the time the plasminogen gets activated, then it's dissolving the clot and making it go away. So it's kind of this, again, kind of delayed activation. It's the same thing that happens here. So we see, again, these different glands that are contributing in different ways to hopefully increase the reproductive success. So, prostate gland, second accessory gland that's going to contributes about 35%, about a third or so. Okay. The last set of accessory glands are the bulbourethral glands. These are also called the Cowper's glands. I don't know, Ernie Cowper or something like that developed or figured them out or something like that. But we tend to call them the bulbourethral glands. These ones dump into, like we saw, just the beginning of the spongy urethra. And these ones contribute maybe a little bit, right, a very small amount to the semen itself. But in fact, they put out a thick, clear mucus that generally is released before ejaculation. So this is helping to kind of clear out the urethra because we know that it's used for urine as well. So again, it's protecting the sperm as they're going to be going through, and also can lubricate the head of the penis itself. So in a way, that kind of contributes to the semen, but again, it kind of comes out as more like coating the urethra instead. Okay, so that's our third set of our accessory glands. So as we've talked about then, semen itself is going to be a mixture of sperm as well as the accessory gland secretions from all of those different glands. So we talked about how it works as a transport medium, right? So it can help to wash things through. It has nutrients in it. It has kind of protective aspects of it, protective and activating aspects of it in terms of enzymes and things like that facilitate their successful pathway and movement towards the potential site of fertilization. We talked about the prostaglandins as well as chemical messengers as part of it too, that can potentially cause peristalsis or rhythmic smooth muscle contractions within the female reproductive tract, which may help to get them towards their site of fertilization as well. Those prostaglandins, interestingly enough, there are secretions that are produced at the cervix. Now the cervix is basically the gateway from the vagina up to the uterus. And so it can facilitate the movement, allow movement to go up, or it can retard or basically prevent the movement going up. And in fact, if you become pregnant, there becomes what's called a cervical plug. So a very, very thick cap, if you will, of mucus that will totally block that, right? Because if we are developing up in the uterus, if we're developing an early embryo and things like that, we don't need any more eggs being released. We don't need any more sperm trying to go all the way up there anymore. We're going to seal that off for a period of time, right? For like 8 months or so while that embryo is developing into a fetus, etc. So the prostaglandins also can decrease the viscosity of the secretions at the cervix. And so that makes it a little bit easier to be able to swim through as opposed to kind of fighting through this especially viscous secretion. When we talk about the female reproductive system, there's also that cycle that we talked about, the ovarian cycle and the uterine cycle. That also affects the mucus that's secreted at the cervix. So at different times in the reproductive cycle, it can be thicker or it can be especially thin, again, helping to allow the sperm to potentially go through.
Is that the same thing that's responsible? So my OBGYN said that if you've been pregnant for a while and you're done being pregnant, and the only scientific way to induce labor naturally is semen, that there's, I don't know if it was a hormone or whatever was in the semen that can activate the cervix to get things started. Is that the prostaglandins?
It's quite possible. I mean, in terms of chemicals, like you were saying, I mean, to a certain extent, it could just be the sexual act, right, is also stimulatory for all of the, not all of it, but a lot of the female reproductive system. So the cervix as well as the vagina around it. So it can increase blood flow, the physical aspect of it can kind of get things moving as well. But the prostaglandins certainly, if that cervix needs to start dilating, start becoming more flexible and start moving, those prostaglandins and those smooth muscle contractions, that can help to kind of chemically induce the process. Yeah. Okay. So there's a few other things. We're just kind of finishing up why there are all these secretions that go into semen in addition to just transporting the sperm. There's actually a hormone in there that is called relaxin that helps to stimulate the sperm so that they increase their motility, because we want them to be fully formed, but we don't need them actively swimming around and bumping into the walls when they're in the seminiferous tubules, when they're in the epididymis, etc. So that hormone helps to increase them so that they're swimming more vigorously as well. The secretions that are put into the semen are also somewhat alkaline, because along the urethra, the male urethra, as well as in the vagina, it can be somewhat acidic, right? Somewhat acidic. We know that acidic secretions are antibacterial, like first line of defense, but we don't want these cells that we have made to kind of succumb to that. And so the alkalinity of the semen helps to neutralize that acidic environment. There are some antibiotics in there as well called seminal plasmin that also help to destroy certain bacteria that may be along that pathway. And then, as we talked about, certain types of enzymes, clotting factors, etc., and liquefying factors that can help to keep the sperm in that pathway and then release them as well. So in an average ejaculation, which can be between 2 to 5 milliliters, there are between 500 to 130 million sperm per milliliter, right? So you're talking, oh, okay, so there's 100 million per milliliter, so you're talking 200 to 500 million sperm per ejaculation. Could be up to 500 million sperm. So as we said, in terms of a reproductive strategy, it's basically just crank out a whole bunch of cells, and a lot of those are going to be imperfect, right? There's going to be some that like don't have tails to them, some that have two heads to them, some that swim in circles, some that don't swim at all. Because if you are a factory and you're really cranking out your product and you're not looking to ensure quality, then what you get at the end is going to be kind of a mishmash of effective cells. And so with all those numbers, we know that some are going to fall prey to a variety of different protective measures along the way. And so we're just kind of doing a numbers game and getting enough out there that hopefully there's going to be some viable ones and active ones that are going to make it up there. Yeah?
I actually have two questions.
Mm-hmm.
So for like, when we're looking at the sperm model and stuff,
Yeah.
is it the DNA, like everything else, like all the sperms pretty much have like the same structure, but it's the DNA that changes? Because like what causes if you have a malfunction sperm that actually does eat the ovaries and actually does make a baby?
Right. So in that case, if we have a malfunctioning sperm, a lot of it has to do with, like we said, like motility issues, etc. So while the sperm, these malformed sperm, could obviously be successfully deposited within the female reproductive tract, into the vagina, they're not going to be able to propel themselves the rest of the way. So if they don't have a head or something like that, then generally they're not going to make it into the cervix. Or if they don't have a flagella, or if they have two flagella, then it's going to be swimming weird pathways and things like that. So the first part of their journey, if you will, is kind of involuntary. They're being washed along, and then they get deposited. But the second part of their journey, they have to be, number one, functional. They have to have all the different components like we saw, the head and the midpiece and the tail, and they have to be actively swimming in order to kind of continue along their journey. Yeah. Second question?
Yeah, it was just like why were like, so like there are like studies where like the baby has different things. So like for instance, like in twins, like when they're joined together, or like one has like the full body but one doesn't have like a leg or an arm.
Yeah.
So I'm like wondering, is it the mom's gene that has that cause, or is it the
That's hard to
sperm?
say. I don't know if that is known. A number of there's a number of things that will genetically determine, right, heritable characteristics about the new individual, right? So there's half the genes that come from the mom, half the genes that come to the dad. But for these different genes that are kind of programmed to create, okay, these ones are going to become a liver, and these ones are going to become the epidermis and things like that. There's a whole sequence of events that has to happen in terms of, okay, now we do need to do mitotic division. Now these cells, now that we have a bunch of cells, they need to become specialized and grow in a certain way so that they become muscle cells and have actin and myosin and stuff like that. And so there's a lot of those chemical signals that tell them when to grow and when to stop growing and what to merge with and things like that. And so if those directions, if you will, those kind of molecular directions at whatever point get off, then again, you don't maybe get a separation where you have things that are grown together that have not fully separated or something like that.
That makes sense. Okay.
So part of it's that, like even kind of relatively simple thing, like in early stages, in early embryonic stages, as we'll talk about later, all vertebrates basically have a tail to them. But depends on what you are, whether you are a fish or a lizard or something like that tail continues to develop. But if you are something more like, right, a human, then that tail is going to actually reabsorb. So there's apoptosis. Early cells have created that area, but then due to stimulation in different ways, they actually start going away instead. And so that there's no, as the development continues, there's not going to be that body part anymore.
Okay.
So, if there's that type of stimulation and it's just that message doesn't get through, then something may still be there, or something may, like we said, be still attached or something like that.
Okay. Yeah, I was just like curious because they're trying to use CRISPR to like fix different stages.
Right. Yeah. So if they determine if there's a genetic disorder that affects development, it can be really significant because, right, if it's already formed, it's kind of like when we talked about hormones like growth hormone. When there's a hyposecretion of growth hormone, if that occurs really early on while things are still growing, that's when you get like gigantism, right? Big effects in the overall body because you've affected this process really early as these cells are still dividing, as opposed to later on when we talk about the acromegaly, when it's just like the facial bones and the hands and the feet that can respond. So if you get some sort of disorder that starts really early, if they can see that there's a gene there that has, that is basically already kind of malfunctioning, then the idea is, okay, if we can catch that early enough, maybe we can insert another gene or something like that, a functional gene that can then guide development in a normal way. So that's what they're working towards, but obviously there's many, many steps before that can be successful. But being able to actually edit genes and things like that is something that's, a new and kind of exciting thing to be able to do, for sure. Yeah, and then a lot of, medical ethics with it as well, right? Okay, so we talked about the production of these sperm cells and how they're going to be transported along these tubes and things like that. But when is that going to happen? So there's a two-step process. One is the internal movement of the sperm along the pathways, which is called emission. And then ejaculation is going to be released from the penis itself. Now the penis, in order to be able to deposit within the vagina, has to become erect, and that's where those channels of erectile tissue come into play. Now we said there's 3 channels. There's 2 corpora cavernosa, and then on the dorsal surface, and then there's 1 corpus spongiosum that encircles the spongy urethra. So what happens is that the autonomic nervous system is basically in control here, so it uses both sympathetic as well as parasympathetic. So in this case, we have some parasympathetic control that is going to allow erection itself, and there's a local chemical messenger that is released that's called nitric oxide. We had mentioned this before when we were talking a little bit about endocrine and chemical messengers. So the nitric oxide allows erectile tissue to fill with blood. These different cavities, there are small arteries, right, going away from the heart that deliver blood to them. It allows those chambers to fill with blood. Now, the interesting thing about it is that running into it, right, we have an artery and a vein, just like usual. We saw that along our pathways. And so the arteries are delivering the blood. The blood's filling the erectile tissue, and it fills it enough so that it actually compresses the vein. So that means the blood can get in during this period of time, but it can't get out. It can't fully circulate. So what does it do? It accumulates. In a way, you think about like edema and stuff like that, fluids accumulating within a tissue. This is blood accumulating within this erectile tissue. So it goes to the tissue, and it can't get out for a period of time. So that maintains the erection in that way. So the corpus spongiosum, as well as the corpus, the other erectile bodies, are going to all fill with blood like that, the cavernosa on top. So now the penis has the erectile tissue that's filled with blood, and the ejaculation will happen as the sympathetic now nervous system is going to cause some of the contraction of pelvic floor muscles as well as squeezing some of these different glands. So just like the gallbladder has smooth muscle on it and we squeeze the gallbladder to release bile, these other glands also can have smooth muscle along their wall, like along the seminal vesicles and things like that. So we're going to transport the sperm from the epididymis up through, with rhythmic peristaltic contractions, up through our vas deferens. The glands are going to dump in their secretions along the way, including seminal vesicles and then the prostate gland. And then, finally, in terms of ejaculation, like we said, pelvic floor muscles, like the bulbospongiosus ones, are going to then do contractions that will finally propel it the rest of the way through the penis itself. So the semen will be ejected from the urethra, deposited into the vagina, and then from there, the sperm are on their own, right? So we've moved them through the internal pathway, gotten them to the external environment, and now they're on their own themselves. So we'll come back to them when we finish up with female reproductive system. We start talking about development, how the sperm and the egg meet. We'll get to that a little bit later. Before, though, we finish up with male reproductive system, we need to remind ourselves a little bit about how this works in terms of cell division as well as fertilization. So way back when, like in 201 when we were talking about like cells, we talked about how there's two types of cell division, right? There's mitosis, and you talked about this in general biology as well. Mitosis and meiosis. And meiosis is the type of cell division where we get sex cells. And in a way, right, mitosis is where you are kind of creating exact copies of a cell, right? So you copy one and you get another one. Holy Cross is looking at me, wondering if I'm limping around after our hike. And in meiosis instead, when this cell division is happening, you basically are tearing a cell in half. Because what you need is what's called a haploid cell, right? In both the male and the female gonads, we're going to be producing haploid cells that have half the genetic material. So in humans, to have a full complement of DNA, you have 46 chromosomes. 23 came from your mom, 23 came from your dad, right? So both the egg and the sperm need to have only 23 chromosomes. Because when they unite, we get 23 plus 23. If we instead did mitotic divisions, the sperm and the egg would both have 46, and you would combine them together, and now you would have an organism that would have like 92 chromosomes. And we know that even having a gene or an extra chromosome can produce all kinds of problems in terms of the developing embryo, right? So let alone having twice the number of chromosomes you're supposed to have. So for meiosis and this gametogenesis, we're producing haploid gametes that when they come together, then form a diploid cell. So we have two copies of all of those chromosomes, two copies of the genes, that gives us our 46. So often they refer to this, a diploid cell, as 2N, and then the haploid cells just are N instead. Okay? Okay. So, we'll get into this a bit. We got 4 minutes. We're going to talk about the spermatogenesis in terms of how this meiosis occurs during the development of the sperm within those seminiferous tubules. So we had talked about how when we look at those seminiferous tubules, what we have are some stem cells that are right along the very edge. So during this process, what we're looking at here, this view that we have right here is a zoomed up view of the seminiferous tubule. So like here's the lumen down here, and here's the wall of the tubule like this. So that's one big donut like that, and the lumen is down here. So this is kind of what we were seeing in the microscope itself. And on the right side, what we can see here are some of the different stages that we were actually seeing in the microscope. So here are the spermatogonia. These are the stem cells. So the spermatogonia, what they do is they do a bit of mitosis and they do a bit of meiosis. So they have to do mitosis so that we don't run out of cells, right? We're going to make cells that will then continue to do meiotic divisions and become sperm, but we have to make new cells all the time so that we don't run out, right? If we just did straight meiosis, after that first division, we'd be out of cells. So there's a lot of mitosis that's going on with these stem cells along the edge, so that we maintain a healthy population of stem cells along the border all the time, these spermatogonia. Then, through the meiotic divisions, we're going to see the development and the halving, right, cut in half, of the genetic material. So we'll see that coming up. But the other thing I want you to notice right now, you see all this yellow stuff here? And this kind of weird shaped nucleus that we see right here? All of that right there, that is a nurse cell. So that is called a sustentacular or a nurse cell. So we didn't mention that when we were looking in the microscope itself, but on either side of that, with the sperm cells developing in the middle, those big kind of Swiss cheese looking yellow things, each one of those things is a cell. It's a nurse cell that, number one, receives the signal from the testosterone when we're supposed to increase our sperm production. That's the receiving cell that has the receptors to receive that signal. Like, we got to continue cranking up sperm cell production. The other thing it does, as you can see, is it creates like a scaffolding. It creates a framework where it can issue and kind of escort the developing sperm cells from the edge in towards the lumen where they're going to be released. So it really forms kind of this internal framework inside of the seminiferous tubules to escort these different cells to their point where they're going to be released. Okay, so we've got spermatogonia, like I mentioned, and then you can see some of our different sperm cells that we were talking about before. Spermatocytes in the middle, but what we're going to talk about next time is that there are primary and secondary spermatocytes. And then when we get towards the inside, we get another stage that's called the spermatid. And notice how the shape is changing, right? We're going from very generic looking round cells with a big nucleus to, number one, more cells, 1 to 2, 2 to 4, because there's 2 rounds of meiotic division. We'll go through that again next time. And then as we get to the spermatid stage is when they're really kind of morphing into this very unique arrow kind of shape that we see down here when we have our spermatozoa or mature sperm cells. Okay? So throughout the meiotic division, what we're going to see is, number one, we're going to see that we're going to be having the genetic material down from 46 to 23, and we're going to see almost like this amplification. We're starting out with 1 cell, and as you can see, we're ending up with 4 cells when we get through our meiotic divisions. Okay? So next time what we'll do is we'll go through that, understand the different steps and stages of spermatogenesis, and then that'll finish up our male reproductive system, and we'll launch into female reproductive systems. What's similar, what's different, etc. Okay? All righty.